This video is going to be a quick overview of the playlist I have on coronavirus and a number of different aspects about its biology. I hope to make another playlist about the effects that this has in humans and varying from patient to patient. So this is more, virus centered. Now, when we look at animals, all right, we classify them into groups, a nested hierarchy of groups, big groups like an order, and then families within the order, and then subgroups. And the members of the orders share features. And so, you can say these are the features which define, this genus, et cetera. We do the same with viruses, even though they're not technically alive. So there is an order known as Nidovirales. This includes lots of viruses which share a common ancestor, including the coronaviruses. They use RNA as their genetic molecule, single-stranded RNA, by the way, and that it is a large genome, which is odd for RNA molecules. As we'll see, the only way that coronaviruses can have so large a genome is that it's able to fix mutations with a proofreading mechanism, something that most RNA viruses lack. They're known as nidovirales because, as we will see, that all members of this group have the same 4 genes for proteins in the same order for the viral structure. So nido literally means nest. And so within the genes, there is a subgenome of 4 nested genes in one particular order. Now, just like the order Carnivora in mammals, not only has the dog family, but the cat family, the bear family, etc., the order Nidovirales not only has the Coronaviridae family with the coronaviruses, but other families which infect other organisms like arthropods, crustaceans, and the like. Now, in a biological family, then you can subdivide the family into genera. Once again, that there was a common ancestor of the family and that new lineages evolved over time. And the same is true of the coronaviruses. This is kind of a simplified version of the family tree, but there is a family tree of this virus family where species diverged and evolved over time, and new species continue to evolve. There are subgroups within the family which include coronaviruses which don't infect humans, the coronavirus MERS, the Middle East respiratory syndrome, which is contagious to camels and humans, and then the coronaviruses, which include not only a few strains which can be among the multiple causes of the common cold, but then also the SARS coronavirus 1 and the SARS coronavirus 2. And so coronavirus did not appear from nowhere. It is part of a family of coronaviruses, which is part of an order of coronaviruses. Now, not much was known about these many of these viruses until the first coronavirus outbreak in 2002. We now recognize that the first cases were known, but it was first identified in 2003, largely confined to that year, although a lab breakout caused a few cases in 2004, and it has not resurfaced since 2004. Now, it seems as if bats were important in this transmission. So coronaviruses infect bats. There's a greater variety in bats than in any other organisms. And so at some point, a virus which infected bats made its way to humans. Now, while that could be possible if humans were in a place where bats were shedding the virus, it's more likely that a bat then gave the virus to another organism like a raccoon dog or a palm civet in Asia where the virus originated. And then it was from these other organisms that coronavirus was then transmitted to the human population. And so one of the videos I have, I won't go through it all, but starts with the timeline of the virus, just to kind of get an idea of just where it originated, how it spread, et cetera. So the first case was November 2002 in Southeast Asia. And then there was an outbreak in cases in February. And then it spread throughout the world. Once again, it started in Southeast Asia, and the majority of cases were there. And in April 2004, a small number of cases associated with a lab were resulted. And so this outbreak started in 2002, as far as we know, and ended in 2004. There were more than 8,000 cases and almost 800 deaths. So that means that the mortality rate was almost 10%, which is very high. There have been no new cases since 2004, and while there were cases in the United States, once again, the fatality rate was 9.6%, 29 cases in the United States, no fatalities. So obviously the hope would be that this virus no longer exists in the world, that no population of bats or raccoon dogs, et cetera, still has it, and that unless there's a new mutant strain of virus, that this is no longer a problem. But in the year 2012, a new strain of coronavirus arose known as MERS, the Middle East respiratory syndrome coronavirus. Bats would have been the ultimate source, but it most frequently is transmitted from camels to humans. Luckily, it doesn't pass from human to human well unless there's close contact. Luckily, it has not spread to large populations. Most of the population, most of the affected individuals have been in the Middle East, although from travel to the Middle East, there were cases which spread to an outbreak in Korea and 2 cases in the United States in the year 2014. Most exposure is through exposure to an infected camel. The problem with this virus is that it has a relatively high mortality rate of about 36%. So if this were something that was in a virus which could spread like the flu, with more than 1/3 of the cases ending in fatality, obviously that would be, a tragic event. But getting back to bats, there are more coronaviruses in bat populations. Now, most of the viruses which would say be in one species are limited to that species because there has to be a relatively precise binding between a receptor on the host cell and proteins on the virus. So then it is not likely that humans would have receptor proteins which are incredibly similar to many other species. So it's not that common that a disease can jump species. Often when it does, the virus quickly undergoes some changes. Now, bats, just their lifestyle makes it easy for viruses to spread. So, for example, if they're huddled together in masses, which they do during the day or which they do when they hibernate, if one of them has a flu-like virus, that makes it easy to spread to other bats. And lots of animals are nocturnal that might be in the same cave or might be in a big dead hollow tree. And so then they are breathing air with little circulation. And so if the bats were shedding a flu-like virus, it would then be breathed in by other nocturnal animals sharing the same space. And if receptors were similar enough, a mutant virus now might be able to jump species and now infect a species other than bats. So the SARS coronavirus 1 migrated from a bat to palm civets and raccoon dogs, and from there migrated into human populations. Now, viruses mutate all the time, and in fact, RNA viruses mutate even more than DNA viruses do, just because the RNA polymerase enzyme, which is replicating RNA, is more error-prone than the DNA polymerases, which have a proofreading capability. In fact, people who study viruses, often are studying mutations because viruses do mutate. But then the problem then is, would a mutant virus be able to infect a new host, or would it be able to, have a different type of infection than the original? The coronavirus can only have a genome as large as it does because it, unlike most RNA viruses, actually has a way of proofreading viruses. Now, one of the problems which we face is in this world where human population continues to grow. When I was born, the world population was around 3.5 billion. That's less than half of what it is today. So more than half of global population is new in my lifetime. But not only does that mean that there's lots more people, there's lots more animals associated with people. Chickens, cows, pigs, and everything else that humans use to feed themselves. And so therefore, as more and more people live on the earth and more animals around people, the odds that a virus might move from another species to humans then goes up in these crowded conditions. And as I said, RNA viruses, they do mutate all the time, but the coronavirus is unusual because it has one of its NSP proteins, the non-structural proteins, certainly one and maybe more, which have a proofreading ability. It's only because of this that the coronavirus can have such a large genome with a large number of genes. Otherwise, the mutation rate would be so large that the virus would degrade and it would no longer be effective at being infectious because of the number of mutations which would accumulate. And so there are a couple of NSPs, certainly NSP 14 and maybe 10, which seem to help the coronaviruses get such a large genome with 27,000 to 32,000 nucleotides in them. Otherwise, the mutation rate would be prohibitive. The viruses make spike proteins, which determine their ability to bind with the receptors on the host cell. Now, you might think, well, can't antibodies then bind to these spike proteins? Well, while they can, maybe that will be effective in the future. The antibodies which have already been produced against SARS coronavirus 1, in case it ever comes back again, are usually not that effective with SARS coronavirus 2. Although these coronaviruses are related to each other and similar, their spike proteins are not 100% identical. They are only 79% identical. And many of the antibodies which would be useful and have been stockpiled in case SARS coronavirus 1 ever comes back as an infectious agent are really not effective here. Even if we did use those antibodies, it's not a permanent fix, because getting antibodies, say in a shot which have been produced, is kind of like getting IgG across the placenta from your mother or in breast milk. It's what's called passive immunity. It might help you in that moment, but your body hasn't learned how to make these antibodies long term. That's active immunity, and that is different. So there is a protein here I indicated in red, the spike protein of the virus, which binds to a specific receptor on human cells. We know what that receptor is. It's called the ACE2 receptor. ACE stands for angiotensin converting enzyme 2. So viruses can enter human cells, and only specific human cells, because the red viral spike protein can bind to the ACE2 receptor. And we know that because in the lab, you can take cells that don't get the virus, and if you could then cause those cells to make the ACE2 receptor, then they do get the virus. And so making the virus, this is a requirement for infection. And so this is the receptor for the coronavirus. As a result, the amount of receptor made can be relevant. So for example, if you had a medical condition such as diabetes, which caused more ACE receptor to be made on your cells, then that would then potentially make your cells more susceptible to coronavirus infection. And I'll hope to pursue that more in the next video. And so this is how the virus enters human cells. But I'd like to just say a couple of words, because this will also then be important in talking about how this affects humans in the next in a later video. So some cells of the body make ACE2, and so therefore would be capable of being infected by the virus. Not only do the cells of the lungs make ACE2, but so do cells of the kidney, small intestine, the heart, and others. There are some cells of the body which definitely do not make this, like those of the nervous system or red blood cells, where there's little or no expression, so they would be less susceptible to the virus. Now, when body cells make proteins, they don't always make the same protein, and so there can be alternate versions of the protein. And as yet, I'm not sure if we understand whether the alternate versions, how significant that is. The version of the lungs is an alternate version of what you find elsewhere. What does ACE do? Well, I'll review this more in the next videos as well. But it's an enzyme that cuts protein, what's called a protease, and it starts at the carboxylic acid end, so it's what's called a carboxy peptidase. So there's this short little signal that we make that can affect blood pressure, can affect vasoconstriction and inflammation. And our ACE enzymes, both ACE and ACE2, can cut a very specific piece off the end. All right, and so here's a peptide chain, a chain of amino acids, and our ACE enzymes make a cut. We have a number of these enzymes, not only in the digestive system, but elsewhere. They serve lots of roles, but they're important. So for example, if you have mice who are mutated and don't make ACE2, they suffer a deterioration of lung function that can be serious. So in the same way, the coronavirus seems to result in less ACE2 in human cells, and that then causes other consequences, which then can result in mortality. And so in the later video that I'm going to make, I'll pursue more what's going on in patients who get this. But just very quickly here, the ACE proteins are part of the system involving what's known as renin and angiotensin. So we have two enzymes. We have ACE, that was the first one discovered, and then ACE2, which is similar. They descended from a common ancestor, but ACE2 is only 40% identical to the first ACE. Coronavirus can infect cells that have ACE2, but not the cells which have the originally discovered form ACE. I have other videos which review this, and it is a little complicated. But for example, if we want to control our blood pressure or fluid volume, the kidneys make a signal renin. This will then cut a protein called angiotensinogen from the liver to make a short little peptide signal called angiotensin 1, which then interacts with ACE to make angiotensin 2. All right. Now, angiotensin 2 has a number of effects. It will, for example, make more inflammation. And in your body, you want to balance stuff. So inflammation up to a certain level is good, but you don't want too much. So ACE1 will break down angiotensin 1 to make angiotensin 2, which promotes inflammation. But we don't want too much inflammation, so ACE2 can then break down angiotensin 2, which promotes inflammation, to angiotensin 1,7, which then results in less inflammation, so it's anti-inflammatory. It promotes vasodilation instead of vasoconstriction, which angiotensin 2 promotes, etc. The 7s, and there's an 8, you can have a 9 in there. That's referring to how many amino acids there are. So if you start off with a chain that has 8 amino acids in it, and you cut it, you can make, angiotensin 1,7, which now has 7 amino acids in it. So in any case, these short little chains are signals for the body for lots of things like inflammation and blood pressure, cell proliferation. And we want a balance. We don't want too much of one or too much of another. And the virus is now going to perturb that balance, because if you take away ACE2, then you're now going to have an excess of the signal which is promoting inflammation, which can now cause damage in the lungs. Once again, that's enough of that topic for now. I'll pick that up again later. And so ACE is the receptor for coronavirus 2, all right? But just a quick note, if we were going to be comparing species, can a virus jump from another species to humans? Well, maybe yes, maybe no. So ACE2 in bats is 81% identical to that of humans, pigs 81%, but it could be a different 81%. And so the virus might not be able to recognize all of the human ACE2 if it's adapted to something else. Now, every virus needs a receptor with which it is interacting. And ACE2 is the receptor for multiple coronaviruses. But as you'll notice, there are different coronaviruses. Some don't infect humans, some cause the common cold, and ACE does not have to be the receptor. There are other potential human proteins which could serve as the receptor for that. Okay. So what happens when the virus binds to the human host cell is that these human receptors, they're enzymes that cut viral proteins. The virus wants that. That's actually one of the triggers which will then activate the virus. So the virus needs a signal before it then, becomes infectious and starts to fuse its membrane with that of a host. And having a spike protein be cut is one of the pieces of that signal. So the ACE2 is an enzyme that cuts proteins. The virus wants that. So when the red spike protein is cut into S1 and S2 fragments, that then triggers the membrane which surrounds the virus to fuse with the membrane of either the human cell, if this is the human cell membrane, or, and it turns out this seems to be the more likely scenario, if the viral particle has been taken into a vesicle, a little membrane bag inside the cell, that this is now fusing with the membrane of the vesicle, All right. So sometimes cells will perform what's called endocytosis. They'll form a membrane bag around something on the outside and bring it in. And so the viral protein can be binding to a receptor either on the cell membrane or inside a vesicle. Now, the ACE2 receptor is involved, but there can also then be other human proteins, other even proteases, which are cutting the viral enzymes, which are also then helping to serve as a trigger. So furin not only is an enzyme which could help contribute to the infectiousness of coronavirus, but it's also required for membrane fusion in other viruses such as HIV, Ebola, and influenza. So human proteins manipulate the viral proteins by, say, cutting them, but the virus is using that as a trigger. And then as a result, the viral membrane, coronavirus is an enveloped virus, which has a membrane around it. The membrane originated from the last human cell that this viral particle had infected. This then allows the virus to fuse with the human membrane. Now, once again, this could be the cell membrane, but it seems that endocytosis, the process where a cell can reach around something that is bound to a receptor and then bring it into an internal membrane bag called a vesicle. This may be a more important route for coronavirus entry. And we think that because the vesicles often can then lower their pH and become more acidic. And it seems that the low pH, the acidity of this vesicle, is another thing which helps to trigger its activity. So rather than fusing with cell membranes, it seems that more are taken into the cell in a vesicle and then fuse that way. So once it fuses, now the RNA molecule is then released now into the human cell. Unlike, say, flu viruses, where the genome may come in pieces, the genome of coronaviruses is all on one big piece of single-stranded RNA. It's what's called positive sense, which I explain in the video, but it is a little more complex. So let me just quickly explain it, and once again, the video will go into greater depth. If something was a negative sense, it means that you could use that as the template to make RNA molecules. So you could use it as a template for transcription. Because this is positive sense, you can, one, have human ribosomes bind to it directly for coronavirus and act as if it is an mRNA, which is now being translated. But then if you want to make more viruses, the virus has to replicate its positive sense strand to make a negative sense strand. And then the negative sense strand is then replicated to make positive sense strands, which then get incorporated into future molecules. So that's what we mean by, positive versus negative sense. And, that was a quick explanation. Obviously, this video will go into it in greater detail. The coronaviruses form a family because they do have similarities. All of them share the last third of their genome, which is larger than what we usually see in RNA viruses, which have the last 4 genes, the S, E, and M and N proteins are all the same, in the same order. All right, so if you were to look at the coronavirus genome, so if each of these nucleotides is representing 3,000 nucleotides. So this is not an RNA genome. It has, say 30,000 bases. So each set of 4 is representing 3,000 nucleotides. The first 2/3 of the RNA genome is coding for 2 proteins. It's called open reading frames because a human ribosome can say, oh, here's a human mRNA. It's wrong. But thinking it's a human RNA, it makes protein number 1, where it binds here and makes protein number 2. And then at the end, in the last third, there are 4 proteins all in the same order for all members of the family, the S, E, M, and N. So that little nest is why we have the nido virales. So there are similarities. But the first 2/3, they can vary because those 2 big proteins are called polyproteins, and they can get broken in all these little pieces. And the little pieces can vary from a cold-causing coronavirus to the coronavirus which causes COVID-19. Okay, and so lots and lots of different proteins here. And in this video, once again, you could go into what all of these proteins do. Some of them are the ones which are replicating the RNA molecule. We're proofreading. We're stabilizing replication. So those big proteins are broken into lots of little pieces, and each would have its own role, some of which are not completely known. But in general, replicating the virus, error proofing it, and a few other things. The S protein, that's the red spike protein, which binds to the human receptor. The E protein forms a part of the envelope that's helping to suppress some of the human immune responses. And the M and N proteins, here's the E protein. It's inhibiting the host stress response here. These proteins may be present in some coronaviruses. But the next couple of phases are going to focus both on the M proteins here in green and the N proteins, which are blue, that you don't really see yet. They will pop up in a future video. The M proteins are going to help the nucleic acid in the middle bind to the membrane and help it curve the right way. So the M proteins are going to have important roles in forming that virus structure. Okay. In host cells, when the coronavirus enters the host cell, it's an enveloped virus. Once again, it has a membrane. This membrane came from the last human cell that it infected. Once it releases its nucleic acid, it's positive sense. So human ribosomes find it and say, oh, this is a human mRNA. Let me make a protein from it. Unfortunately, it's the viral positive sense molecule, and thus then the human ribosome then makes the viral proteins, those big polyproteins, which then get cut into little pieces. So the coronavirus RNA actually looks like a human mRNA. Okay. The virus, those polyproteins will make more virus. So let's say one viral RNA molecule gets inside the human cell. Those proteins which were made, they will recognize the positive sense. They will then replicate that to make negative sense strands, lots of them. And then those negative sense strands can then be the template for more positive sense strands, which can make more viral particles, or can be the kind of the mRNAs from which more viral proteins are made. Okay. So as more of the viral nucleic acid is made, the virus, if it's going to make more viral particles, which can then infect future cells, at some point you have to complete the structure of the virus. And so here are those proteins which are being made, and they are now being incorporated into human membranes. So inside the cell, there's lots of membranes, and now these viral proteins are ending up in the human membranes. But then there is what is called the N protein. This will now bind the RNA of the coronavirus genome. So RNA molecules can fold upon themselves. The N protein acts as what's called a chaperone protein, which helps the viral protein to fold in just the right way. And so we don't want the virus doesn't want its protein to fold in the wrong way. And so the N protein causes it to fold in the right way, making these loops. And then the RNA with the N protein in the middle of the viral particle can then bind to the green M protein in the membrane. And this now allows the viral nucleic acid to be packaged inside a larger particle. It's now ready to infect the next cell. So the M protein in green is the most abundant protein in the coronavirus particle, outnumbering the blue N proteins by a ratio of 1.4 to 1. Once again, the N protein binds to the RNA genome, all right, and then helps it to form the right structures so that now it can be packaged by having the N protein on the inside bind to the M protein in the membrane itself. And so now this will form a virion inside a membrane, and this is actually happening inside the human cells. So human cells have lots of different kinds of membranes. The rough endoplasmic reticulum can make vesicles, which then go to the ER to Golgi intermediate compartment, which can then go to the Golgi. So there's all of these membranes here, the RER, the ER to Golgi intermediate compartment, the Golgi, and notice there are vesicles going back and forth. And so lots of processing occurs in normal human cells of proteins. But this is also where the virus is being built. It's here that the viral proteins get added to human membranes. It is here that the N protein binds to the M protein. And so that now a virus is now made. And now this membrane can now bud from the human cell. So while some viral particles are lytic and cause the host cell to burst, these viral particles will leave one at a time. Once they've made these membrane bound virions, they will now bud from the host cell. So that's a little bit about the biology of coronaviruses and coronavirus activity, but now focusing on the host response and why some people might be more susceptible than others. That is what I'll put together in the next video.